In this video, we're going to be looking at blood and how blood is a liquid tissue. Because remember, by definition, tissues are groups of cells that work together. And in blood, we have what we call the formed elements, which are the different types of cells that make it up. We have the erythrocytes, which are your red blood cells. You can kind of think of them as baskets. Their job is to transport. We have leukocytes, which are your white blood cells. I call them your police department because they're your immune system. And then we have these things called platelets, which I call the plugs, because they are what make up your blood clotting. As blood, it is loose connective tissue, so we do have to talk about its matrix, which is a liquid, and the ground substances, which would be things such as plasma. Now, with this video, we're going to focus mostly on your erythrocytes, which are your red blood cells. And as you can see, remember, structure determines function. These cells look a little bit like boats because their main job is to transport materials and nutrition. We talked about how they are red. And they're red due to the presence of a protein called hemoglobin. Anytime you see hemo, that's going to refer to blood, and globin is a protein. Now, we talked about how proteins have different levels, and so the highest level you can have is what's called a quaternary protein. Hemoglobin is actually 4 proteins that all work together to act like one big super protein. And therefore, one hemoglobin can hold 4 oxygens at one time. And one red blood cell holds a lot of hemoglobin. So think of red blood cells as baskets or bowls or boats, and because of their shape, remember, folds equal surface area. Since they're folded, they can carry all of this hemoglobin so that they can carry all of the oxygen. They are anucleated, and I don't like the way that this definition is stated. Your red blood cells are made in your bone marrow. They're made by your red bone marrow, and they're made using a protein in hematopoiesis. Hematopoiesis, remember, is the making of blood, called erythropoietin. Remember, anytime you see poet means that you're creating something. So erythropoietin is a hormone that tells your red bone marrow to create red blood cells. Well, when the cells are made, they are round, just like what you think of as a cell, and they have a nucleus. But we have a special type of capillary in our bone marrow. We're going to talk about this later. It's called sinusoidal capillaries. And your red blood cells have to kind of squeeze their way into that capillary. So imagine a room with a very, very small door, and there's thousands of you all trying to squeeze through that little door. Well, that's what happens when red blood cells get made. They have to squeeze into a little door, and in the process, their nucleus gets damaged. I don't like how it talks about how the nucleus pops out. If the nucleus were to pop out, the cell would have exploded. But in the process of squeezing into the blood vessel, the nucleus gets damaged. And the nucleus gets damaged, it falls apart. And without another circle inside of your cell, your red blood cells do cave in. So anucleated means they don't have a nucleus. But because they don't have a nucleus, they do fold. And again, folds are good because folds equal surface area. So we call these biconcave discs. They basically look like inner tubes, but they don't have a hole that goes all the way through them. And so this gives them their structure determines function. So it caves in where the nucleus was once found. But this, again, allows them to act like boats. It allows them to act like baskets so that your red blood cells are able to carry things through your body. And because they don't have a nucleus, they cannot go through mitosis. And they cannot create new proteins. Remember, you have to have DNA in order to do DNA replication and to do transcription and translation. When the nucleus gets damaged, they're not able to create a lot of their own proteins. So your red blood cells, on average, one red blood cell lives about 120 days. So your red bone marrow is responsible for constantly making new red blood cells. Now, after about 120 days, your red blood cells will begin to biodecay. Now, lucky for us, not all of our red blood cells biodecay at the same time, but they do begin to break down. And when things begin to break down, they can form acidic environments. That's why you've all heard the stories about old graveyards, how they're all spooky because things won't grow there. Well, remember, back in the day, when we buried people, we buried them in wood boxes or we just wrapped them in sheets. Well, when things decay, hate to say it, the juices that are given off are acidic, and so that can cause the soil to become acidic, which means nothing's going to grow there. Well, when our red blood cells begin to break down, they can form an acidic environment. So we do have this little organ called the spleen. And the spleen is found near the stomach. It's actually kind of wrapped up in your intestines. And its main job is to filter out decaying erythrocytes. Remember, erythrocytes are your red blood cells. Okay. Since RBCs, red blood cells, don't have a nucleus, they begin to decay after about 128 days. And when things decay, they give off hydrogen ions, and hydrogen ions can change the pH of a solution. And they can begin to make your bloodstream acidic. So we need our spleen in order to filter this out. Okay. Now, iron is very precious to our body. So as the spleen is filtering out the red blood cells, hemoglobin will be broken down. And it's broken down into iron, which will then be recycled, and something called bilirubin. Do not get bilirubin mixed up with bile. Bile is what's made by our liver and stored in our gallbladder to help our bodies break down fats as they pass through our intestines. Bilirubin is a waste product that comes from the breaking down of red blood cells. The bilirubin gets added into eventually our fecal material, and this is what basically causes the brown color of our feces. Now, go back here with red blood cells. Okay. When we talk about abnormal red blood cells, these are obviously red blood cells that don't have the correct shape. As you can see in this picture, it's supposed to be like this flat inner tube. It's supposed to have these folds where the nucleus was destroyed and it kind of caved in on itself. It's supposed to have this biconcave shape. And again, structure determines function. By having this biconcave shape, it has folds. And folds equal surface area. So the more folds it has, it basically becomes a basket and allows it to carry things. Well, when red blood cells are first made, they are round. And like I mentioned, when they have to squeeze through that door to get into those capillaries, the nucleus can be damaged. Well, however, not always does this happen. And so sometimes a red blood cell that's round gets into our bloodstream. If you were to prick your finger and take a drop of blood and look underneath the microscope, it's very likely you would find a perfectly round red blood cell. These are what are called ovulocytes. Now, these are red blood cells. They are made normally. It's just when they entered the bloodstream, they got lucky. Their nucleus was not damaged, so they're still perfectly round. So they never actually lost their nucleus. However, since they don't have these folds, they don't have the same amount of surface area. So they're not really baskets, they're not boats. They are not able to perform their jobs the way that they're supposed to. Without that little concavity, they can't carry as much. So they basically, even though they're perfectly round and like a cell's supposed to be, and they have nucleus and DNA like a cell's supposed to, it doesn't function the way that our bodies need red blood cells to function. So a lot of times these will be destroyed. Like I said, it's very common to have a drop of blood that has this in them, so it's nothing to freak out about, but this is not how our bodies have adapted. Now, you've all heard of sickle cell anemia, and sickle cell anemia is a genetic disorder in which the red blood cells change shape. Remember, structure determines function. So if you change the shape of something, it's not going to work the same way. So in the case of sickle cells, this is a genetic mutation which causes the cells to basically change shape and they become very hard. And sickle means C-shaped, and that's what you're seeing here in this picture. You have your normal red blood cell and you have your sickle cell red blood cell. And with the sickle cell anemia, since they're not shaped like baskets, they're not going to have the hemoglobin that a normal red blood cell would have. So they're not able to carry the same amount of oxygen and nutrients and waste products that a red blood cell would have. And at the same time, our red blood cells, they're like inner tubes. They're flexible, they're bounceable, but sickle cells are very, very, very hard. So they have a hard time getting through our blood vessels. They can't flex, they can't change shape. Remember, we go from really big blood vessels to smaller blood vessels to smaller blood vessels. It's like going from a 4-lane road to a 2-lane road to a 1-lane road. So our red blood cells are able to flex and change shape, but sickle cells don't have that ability. And based on the way that they shrink, you can see from this picture, they do form sharp points on the ends. So it is possible that as these sickle cells pass through blood vessels, it can cut the inside of those blood vessels. And yes, blood vessels can bleed. So you could cut those blood vessels so that they don't work properly. So a lot of people with sickle cell anemia have to be careful with bruising because, they'll just have these bruises that show up. And a bruise, remember, is a popped blood vessel under the skin. So you're bleeding under the skin, the blood doesn't come to the top. It's incredibly painful because you do have these blood cells scraping through your blood vessels. And there's also what are called teardrop cells. Teardrop cells are seen in people who are carriers for sickle cell anemia. So it's kind of like, if you were to draw the perfect little teardrop, it's got a point at one end, but then it's nice and round at the other. These are red blood cells that seem to be trying to change shape, but they realize, oh, no, I'm not supposed to be changing shape. So they form this little teardrop. And remember, in most cases, nature has gotten rid of genetic disorders such as this. It is a rare, recessive, actually I believe it's codominant genetic disorder. But the reason that this still exists in nature is because people with sickle cell anemia are basically immune to blood disorders such as malaria. Sickle cell anemia was believed to have started in Africa. If you were to look at native Africans, over 50% of all native Africans are carrying the gene for sickle cell anemia. It makes them immune to malaria. Malaria is a protist that gets into our blood, and people with these hard sickle cells, the plasmodium can't affect these cells, which makes them immune to malaria. So what's believed to have happened is that at some point in time, somebody in Africa was born with a mutation. Remember, mutations are what cause evolution, and this mutation caused them to have these sickle cell shaped cells. And this person was able to survive, and this person passed this trait on to their children. And again, remember, this is just theoretically what believed to have happened. Well, over time, remember, Africa was a tribal, and in some parts of Africa, it's still a tribal, small, close-knit communities. So over time, people who lived in this tribe were at least carriers of sickle cell anemia. And what's believed to have happened is as they were traded off, which did happen in African tribes, or these arranged marriages, that this trait got passed into other tribes, or it's very possible that a malaria outbreak took place, and the only people who survived this outbreak were of the same family who were carrying this trait. So they had to leave their tribe and go to other tribes in order to find mates. And over time, this trait became predominant. Well, unfortunately, when slavery took place, people from Africa were brought to other parts of the world, and this is how this condition began to spread. It is still prevalently dominant in people of African descent. However, it is showing up. Thankfully for us, we don't have the same social economical setbacks that our ancestors did. And we are beginning to see sickle cell anemia show up in other ethnicities due to the fact that we do have multiracial relationships now. It's also showing up in large numbers in people of Mediterranean descent. Not really sure why. That's one of the things that scientists are studying. But this is one of those traits that nature is kind of hesitant to get rid of because it does serve a purpose as far as malaria is concerned. Okay. Now, we can also have what are called crenated cells. The term crenation means to shrink or to shrivel. And crenated cells are seen when there's oxygen starvation. Now, unfortunately, you've all heard of people who seem to have died in their sleep. Well, one of the first things that they're going to do, if they don't perform an autopsy, they're still going to take blood samples because they're going to be looking for things such as crenated cells. If cells are crenated, that means that that person was suffocated. They might have unfortunately, they might have been murdered by somebody putting a pillow over their face, or just the way that they were twisting and turning in the night, they might have somehow suffocated themselves. This is what a lot of doctors, you guys have all heard of sudden infant death syndrome, where unfortunately infants just seem to die in their sleep. Well, this is one of the things that doctors look for because they're wanting to see, did the child suffocate somehow? Did they get wrapped up in their blanket? Did they get wrapped up in their sheet? So unfortunately, this does occur, but this is what those scientists are looking for in those situations. Now, like I mentioned, we've talked about those are the shapes that you can have of red blood cells. When we talk about disorders, remember disorders tend to be genetic. If it's a disease, you catch it, like malaria. If it's a disorder, it's due to a genetic mutation of some type. And so we did mention sickle cell anemia and how this is that genetic disorder. And this is a picture of sickle cell anemia. You can see what your normal red blood cells are supposed to look like. It's supposed to look like nice little baskets that can carry things. And you can see how the sickle cells kind of shrivel up. They're not able to carry as much. So people with sickle cell anemia a lot of times have a hard time, how can I put this? They have a hard time repairing their tissues. You know, cuts take a long time to heal because they can't carry nutrients. They become out of breath very easily because their bodies aren't able to carry that oxygen. But anemia is a term that you've heard of a lot. And by definition, anemia means iron poor blood. So people with sickle cell anemia are going to have iron poor blood because their red blood cells can't carry the iron and the hemoglobin like they're supposed to. So with anemia, anemia can be environmental. Anemia can be genetic. So for example, I have genetic anemia, and just for some reason, my body does not recognize the fact that iron is available. So my body kind of ignores the iron, so my body has a harder time making hemoglobin. It can be environmental. It can be from the foods that you eat. You're just not getting the iron that you need. Ladies, you can become anemic about once a month, right before your period starts. But with anemia, for some reason, you don't have the hemoglobin. And if you don't have the hemoglobin, you can't carry the oxygen that you need. And a lot of us have, males and females, wear gold jewelry. And sometimes you might notice that gold jewelry turns your skin black. Not green. If it's green, it's not real, but black. So you, ladies or gentlemen, you might have earrings in and you take your earrings out and there's like a black residue on your skin, or you might have a ring and it just gets like this black residue around it. Well, just a little jewelry fact. As you know, most of the gold is not pure gold. It's gold mixed with other chemicals. And people who are anemic, sometimes our skin becomes very acidic, which is why people who are anemic tend to have dry, rough skin. And ladies, again, this is why right before your period, you might notice that your skin becomes drier. Well, the skin becomes acidic and the acid begins to react with the gold, excuse me, and it forms, excuse me, it forms an ash on your skin, and that's that black color. So let me see if I can get this camera to turn on here. Nope, camera's not going to work. So I'll have to show you guys later about how I can take a gold earring or a gold ring and write on my skin with it and basically have a black ash tattoo. But that's why your skin turns black. It basically means that your skin is acidic, and it's probably because you are anemic. And again, anemia can be environmental. It could be something that, you know, you haven't had a lot of iron in your food, or ladies, it might be right before your menstrual cycle. It happens to all of us. Now, polycythemia, poly meaning many. Polycythemia is when you can have too many red blood cells. And you're like, oh my gosh, why is having too many red blood cells a bad thing? Because, you know, if red blood cells carry nutrients and waste products for me, I want a lot of red blood cells. Well, in the case of polycythemia, remember, compare blood, pancake syrup versus honey. You know, if you eat pancake syrup, it's sweet because it has sugar in it. But if you put pancake syrup in a spoon and turn the spoon over, the pancake syrup flows very easily. But if you do that with honey, it takes a long time for the honey to begin to flow. Well, the same is true with our bloodstream. We don't want our bloodstream to be too thick. So if you have too many red blood cells, your blood has a harder time flowing. And so this is a genetic disorder. It can be environmental. For example, if you were to, this is why for people who climb Mount Everest, they have to go up in stages because their bodies have to adapt. If you go to Denver, Colorado, the Mile High City, when you first get there, it's like exhausting. Your body in oxygen poor situations will realize, oh my gosh, I need more red blood cells. And your body will begin to speed up and make more red blood cells. However, our blood vessels aren't adapted to having more red blood cells, and so our blood gets thicker. People who live near Mount Everest, the native people there who always act as tour guides, who help people go up and down Mount Everest, they've adapted to that environment. And if you compare my blood vessels to their blood vessels, the diameter of their blood vessels was actually larger. So they have more red blood cells, but through evolution and through mutations, their blood vessels are bigger. So they don't have this same problem. And then there's that sickle cell anemia again. Now, you've all heard of blood type. And if you remember when we talked about cells, we said that all cells have proteins on the outside of them. And we have this thing called a marker protein, which marks that cell as yours. But then you also have proteins that say, hey, I'm a red blood cell, or hey, I'm a brain cell, or hey, I'm a white blood cell. So proteins can be used as markers to say, this is what kind of cell your body has. So no matter what creature you are, a red blood cell is a red blood cell. So red blood cells will have these proteins on them that say, hey, I'm a red blood cell. So my red blood cells have the same red blood cell proteins on them as your red blood cells. That's what makes them a red blood cell. And people have been doing blood transfusions for years. There's actually hieroglyphics in ancient Egyptian buildings that show what we believe to be blood transfusions. And sometimes blood transfusions worked, and sometimes blood transfusions didn't. Now, you and I all know that we have marker proteins. So if you get a blood transfusion, it's not your blood. So your immune system will destroy it. But at least you have blood that you can use until your body can make more blood. But we learned that through the study of transfusions and basically, unfortunately, trial and error. And sometimes when people did blood transfusions, they worked. But sometimes when people did blood transfusions, they died. So the question was, what is going on if blood is blood? So they began to study blood cells, and a gentleman named Landsteiner is the gentleman who was able to name and come up with the fact that we have 4 different types of blood. You guys know that we have A blood, B blood, O blood, and AB blood. Well, what that's referring to is what kind of proteins do you have on your red blood cell? Now, what that means is when you look at red blood cells, all red blood cells are going to have the same protein on them. And that's what says, hey, I'm a red blood cell. But when Dr. Landsteiner was studying red blood cells, he noticed that sometimes there was an extra protein on it. And on here, you'll see these things called antigens, which is not a term that we've used before. An antigen is a protein on a cell. So our marker proteins are antigens. Okay? The protein that says, hey, I'm a red blood cell is an antigen. So every cell in nature, even non-cells like viruses, have proteins on their surface. So these are what we call antigens. So if you're a red blood cell, you have the RBC antigen. Me, you, your dog, a mosquito. If we have red blood cells, we all have the same protein, the same antigen, because that's what makes it a red blood cell. But Dr. Landsteiner discovered that some people had an extra protein. They had the antigen that said, hey, I'm a red blood cell, but some of them had an extra protein. And I always draw these as shapes, and you can kind of see this in this picture. Some of the blood cells have what look like pointy proteins on them, and some of the red blood cells have what look like white or like round proteins on them. Well, when he was looking at red blood cells, he noticed that some people had this extra protein. And in this picture, it's the pointy protein. And since he discovered this extra protein first, he called this protein A. So if your red blood cells have this extra protein on it, you have A blood. Now, this protein is dominant. So if you remember genotypes from biology, you could be big A, or you could be big A, O, which stands for 0. Well, in his studies, he noticed that some people had a different extra protein. And in this picture, it's that round protein. And since he discovered this one second, he called this his B protein. And so again, with your genotype, you could be big B, or you could be big B, O. B is dominant. He noticed that in nature, some people had no extra protein. So they had 0 extra proteins. And this is how O blood got its name. The O is actually a 0. you have 0 extra proteins on your red blood cells. This is recessive, so genetically you're little, or 00 or 00. And a very, very rare few had both extra proteins on it. This is a codominant situation, and this is what we call AB blood. A is dominant, B is dominant, O is recessive. So all of these cells have the protein on them that says, hey, I'm a red blood cell. But it's possible that you have an extra protein on you. And that's what gives us our A or B. If you have both extra proteins, you're AB, or if you made 0 extra proteins, you have O blood. Which leads us to our blood typing chart. In the case of type A can donate to A or AB. And you're like, okay, well, what's going on? And I kind of skipped ahead. Antigens are proteins that you have on the surface of the cell. Don't get antigens mixed up with antibodies. Antibodies are proteins that we use as weapons to fight off a bad guy. And basically a bad guy is any cell that doesn't have our marker protein on it. So if you have A blood, you're going to make a B antibody. If you have A blood, you're going to fight off the Bs. If you have B blood, you're going to make an A antibody. You're going to fight off the As. If you have AB blood, you're not going to make any antibody because you don't want to fight off yourself because you have A and you have B. If you make O blood, you're going to make both A and B antibodies to try and fight off everything. So going with that, if you have type A blood, think of it this way. If A blood was red water, if you add red to red, it's still red. There's no reaction. When the wrong kinds of blood mix together, that's what's called agglutination. And this is what was happening with people with blood transfusions. If they were just randomly putting blood together. Well, if you just happen to have A blood and you just happen to get A blood, nothing happened. But if you had A blood and somebody gave you B blood, you would form blood clots, which is what's called agglutination. So what would happen is all the A blood would kind of clump together, and then all the B blood would kind of clump together. So with A blood, A can donate to A because red water and red water is still red water. Well, think of B blood as being blue. So obviously AB blood, when you mix red and blue together, is purple. Well, if I have a beaker of purple water and I add a little bit of red to it, it's still purple. So A can give to AB without any reaction. But notice A can get from A, but A cannot get from AB. If A blood was a red water, if I added purple water to the red water, it turns purple. So even though A can give to AB, AB cannot give to A. O would be like plain water. If I have A blood, which is red water, and I add some plain water to it, it's still red. So type A can donate to A or AB, but it can receive from A or O. Likewise, B blood can donate to B or AB. If you think of B blood as blue, B blood and B blood, blue and blue is still blue. If you think of AB as purple, if I have a big beaker of purple water and I add some blue to it, it's still going to be purple. But it can receive from B or O. Now, AB blood is called the universal recipient because it can get blood from anybody. However, it can only give to AB. Okay. So again, think of AB blood as a beaker of purple water. If I add red to it, which is A, it's still purple. If I add blue to it, which is B, it's still purple. If I add clear water to it, which is O, it's still purple. There's no reaction, so it's safe. However, if I give somebody with A blood, which is red, purple water, there is a color change. That's a reaction. That's bad. So AB is the universal recipient. It can get from anybody, but it can only give to somebody with AB blood. The opposite of that is O blood. O is called the universal donor. It can give to anybody. It's clear water, but it can only get O. So if you have O blood, it'd be like you having a beaker of clear water. And I have A blood, so I would be adding red water to your white water, that's a color change, that's your reaction, that's not good. So this is what it means to have A blood, B blood, AB blood, or O blood. Now the positive is something totally different. The positive is what's called the Rh factor. Now we knew about A blood, B blood, AB blood, and O blood. But sometimes when you took two A bloods and mixed them together, there was still a little bit of clumping. And the question is why? If they're both A, why am I having this, what's called agglutination? And so they were studying rhesus monkeys, which are kind of close relatives to ours. And when studying their blood, they noticed another protein that you could have. And they called this the Rh protein, named after the rhesus monkeys. Okay? If you have the protein, you are Rh positive. And this is actually dominant in nature. Most humans, I'd say about 80% of all humans are positive. If you're missing that hormone, you're Rh negative. So that's what it means to be A positive or A negative. And in most cases, it doesn't really play a role unless you have to worry about a blood transfusion, or ladies, you're going to have a baby. Again, about 80–85% of the population is Rh positive, so that is the dominant trait. Well, remember, when you are pregnant or when you are a fetus, you should have no contact with your mother. You make everything for yourself. You made your placenta, you made your umbilical cord, you made the membranes that surround you, you made the fluid that you're floating in. Because you guys now know about marker proteins. And you know that marker proteins mark your cell. Well, a fetus's marker proteins are different from the mother's marker proteins. So therefore, the fetus really should have no contact with the mom. That's why the fetus makes its own membranes, its own fluid. It makes everything for itself. Well, a lot of times, most people, if you think about it, don't know their blood type. Back in the day when you were a baby, they used to prick your heel and tell you what blood type you were. So for example, I know I'm A positive. But when my daughter was born, I asked what their blood type was. And they said, well, would you like us to do a test for you? I'm like, sure. And they're like, well, it's going to be about $500 because insurance no longer pays for it because it's not considered a medical necessity. But with genetics, you can kind of figure out what your blood type is. And honestly, if you ask your doctor, they might be able to tell you. But a lot of women, when you get pregnant, they'll go and they'll do all these blood tests on you. But they know your blood type. And I can honestly say, I don't remember them ever asking me what my husband's blood type was when I was pregnant. What they are looking for is, I guess with blood type, are you positive or are you negative? And mom, if you're positive, there's a very good chance that your child will be positive as well. But if the mother turns up being negative, which is recessive, they're going to put the mom on an autoimmune blocking drug for the duration of her pregnancy. Because there's something called erythroblastosis fetalis. Again, a big word, but break it down. Fetalis means it's a fetus. Blast means to make, and erythro is referring to red blood cells. So this disorder, this disease, this condition has something to do with the fetus making red blood cells. Well, what can happen in a mother who is Rh negative? She is recessive. Recessive is less common in nature. If a mom is Rh negative, the doctors are going to assume that her child is Rh positive. Because positive is dominant in nature, they're going to assume that the dad is dominant, unless they know for a fact that the mom is negative and the dad is negative, which they usually don't. Okay? They will put the mom on an immune suppressing drug for the duration of her pregnancy. Like, I know my mom is negative. So as soon as she was pregnant and they realized she was pregnant, they put her on a medication because I'm A positive. My mom is A negative. But they don't know my blood type until I'm born. But what can happen in this situation, if the mom is negative and the child is positive, if by some freak of nature there is fluid mixture or blood mixture, the mom's immune system can destroy the fetus's ability to make red blood cells. That's what that term means. So as soon as the child is born, the mom can stop taking this immunosuppressant drug.